The sequence of amino acids in a peptide can be analyzed using a variety of techniques, and one of the ways that we do that is called the Edman degradation. So if you're wondering what's the sequence or what is sequencing a peptide, well, let's say you have something like leucine and enkephalin that I put here, okay? So the order that the amino acids are in, the tyrosine, glycine, phenylalanine, leucine, how do you determine the structure of that, or how do you determine the order in which the amino acids are arranged in this pentapeptide or a bigger peptide or a protein? And that's what sequencing is. So Edman degradation involves removing one amino acid residue at a time, and then you can identify each residue as it's removed. The process is repeated until the entire peptide has been sequenced. And to perform an Edman degradation, a peptide is first treated with phenyl isothiocyanate followed by trifluoroacetic acid. And these two reagents remove the amino acid residue at the N terminus and convert it into what's called a phenylthiohydantoin intermediate. So here's our peptide down here, okay? And you can see that it's turned around because the N terminus is on the right-hand side now, but this is just to, illustrate it in a reaction. So the first thing we do is we treat it with the phenylthiohydantoin, and that makes, and then TFA, trifluoroacetic acid, and that removes one amino acid. So you see that the part here that's highlighted in blue, our last amino acid, is removed, and it's converted into this phenylthiohydantoin intermediate. Well, there's only 20 amino acids, so there's only 20 of these derivatives that you're going to form. Now, the phenylthiohydantoin derivative can be analyzed using NMR or mass spec very quickly. And the process is automated and can be repeated until all the amino acids in a peptide have been identified. So it says here you can go up to 50 amino acids. So a very clever way, picking off one amino acid at a time and then elucidating the structure, and thus you can sequence the peptide. Now the mechanism of Edman degradation is shown here. I'm not going to ask you this mechanism. You should just know that you form the phenylthiohydantoin derivative, and then you can elucidate the structure of that using mass spec or NMR. For peptides that have more than 50 residues, the removal of each residue one at a time isn't practical because unwanted side products can accumulate and interfere with the results. So larger peptides, what they do is they first analyze them by cleaving them into smaller fragments, and then they sequence the fragments. So they break it up into little pieces, and then they analyze the pieces. A variety of enzymes called peptidases selectively hydrolyze specific peptide bonds. You'll learn more about those in biochemistry, but in this class we only cover a couple of them. For example, trypsin is a digestive enzyme that catalyzes the hydrolysis of a peptide bond at the carboxyl side of the basic amino acids arginine and lysine. And so since you know that it goes N-C-carbonyl, right, it's going to cleave on the carboxyl end. So you see here we have lysine and we have arginine. And so it's going to cleave here and here. And then you're going to end up with these fragments, and then you can go and sequence the fragments using your Edman degradation. Chymotrypsin is another digestive enzyme which selectively hydrolyzes the carboxyl end of amino acids containing aromatic side chains, which includes phenylalanine, tyrosine, and tryptophan. So again, carboxyl side, and you can see here we've got a phenylalanine, we've got a tyrosine, and a tryptophan. And so we end up with 4 fragments in this case. And again, we analyze these fragments using the Edman degradation. And so you probably noticed that what we can do is we can take the fragments from the previous slide and the fragments from this slide, and then we can assemble it and figure out what the sequence of the peptide was. Okay, so a peptide with 22 amino acid residues is treated with trypsin to give 4 fragments, while treatment with chymotrypsin yields 6 fragments. Identify the sequence of the 22 amino acid residues in the starting peptide. So if we start with the trypsin, remember trypsin is going to cleave arginine on the carboxyl end and lysine. And so that tells us that valine, this one here, this must be the C terminus. So we're able to figure out what the C terminus is very quickly in this case. And then if we look at the chymotrypsin fragments, okay, so we have tryptophan, we've got a tyrosine, another phenylalanine, a phenylalanine, a phenylalanine, and hey, what do you know? Valine again, okay? So you can clearly see that the C terminus is the valine. And what we're going to do is we're going to go backwards. Okay, so we're going to go in reverse. So we know that this is the C terminus and this is the C terminus. And you can see that it goes valine and alanine. So that's for sure. Okay, so we're going to start and I'm going to go backwards. So we're going to go valine and then backwards, alanine. Okay, so we've got rid of those. Then we go phenylalanine, proline, glycine. So phenylalanine, proline, glycine. Okay, now you're not sure which one of these three you have to go with next. So what you're going to do is you're going to look for phenylalanine, proline, glycine and the other ones. And you see it's right here, phenylalanine, proline, glycine. So now we have arginine, cysteine, methionine. So arginine, cysteine, methionine. So we cross all those off. And we see that right here, arginine, cysteine, methionine. So we know that before that it must be phenylalanine, histidine, and tryptophan. So phenylalanine, histidine, tryptophan. So we look for those. So we see phenylalanine, histidine, and tryptophan is here. And this is the only place we see tryptophan in the whole thing. So before the tryptophan, so I'm going to go backwards. So I'm running out of space here. So then we go to arginine, leucine, isoleucine, valine, proline, lysine. So let's scribble those down. So arginine, leucine, isoleucine, valine, proline, and lysine. Okay. And so we see those ones right here as well. Leucine, isoleucine, valine, proline. Then the only thing that can come next are these five, right? Lysine, lysine. So then we have our tyrosine, alanine. So valine, and those are these three here. So lysine. So we have tyrosine, alanine, and then we have valine. Sorry, valine, I forgot to put that one in here. And the last two, again, I'm running out of space here. But then after the valine, we have phenylalanine, methionine. So phenylalanine, methionine. Okay, so you might want to look at the solutions manual for it. Drawn it a little bit neater, okay? But this is the sequence of the amino acids. Anyhow, so there you go.